Dear students of Dentistry faculty, welcome to the second lecture on the digestive system. I am Günel Əyyubova, Senior Lecturer of Department of Histology, Embryology and Cytology of Azerbaijan Medical University. Today I will talk you about the alimentary canal. We will focus on the general morphofunctional features of alimentary canal, the esophagus, stomach, small and large intestines. We know that the alimentary canal, it is tubular portion of digestive system. Here taking place here the food will liquefied, chewed, digested. Nutritional components and the water from the food will be absorbed and non-digestible components will be eliminated. The alimentary canal starts in the oral cavity, it continues into the esophagus, pharynx, then esophagus, stomach, then small and large intestines. In the wall of the alimentary canal, we can distinguish several components. These are several layers. That is a histology of the alimentary canal. Innermost coat, here the mucosa. So innermost coat of the alimentary canal, that is a mucosa. Mucosa includes three main components. First of all, epithelium. Epithelium may be different types. We will talk in each of these components. Then under the epithelium, we can see the lamina propria. Lamina propria is a loose collagenous connective tissue that may include glands, and it contains lymphatic nodules and lymphatic vessels. Under the lamina propria, we can see lamina muscularis or muscularis mucosa. That is also a component of the mucosa, and it includes mainly smooth muscle cells that provide mobility of the mucosa. Under the mucosa, we will see the submucosa here and also in esophagus, and here we are looking at the small intestine structure. Then we are looking at the large intestines. All of them will have same general principle of structural organization, but in each of these components it will have its own specializations and modifications. So the submucosa, we stopped in the submucosa. Let's look here, that is submucosa here, and this one by more whitish colored. Submucosa include dense irregular connective tissue. It includes plexuses of vessels, blood vessels, lymphatic vessels, also nerve plexuses. In esophagus and in duodenum, we will see also glands inside of this submucosa. Then we have muscularis externa, that is muscle coat. It mainly includes inner circular and outer longitudinal layers, but in stomach we will see three layers. Then we are coming to the outermost coat. Outermost coat of the alimentary canal, it is connective tissue that may be or may be not covered by simple squamous epithelium, which is called mesothelium. It depends on the organ. If the organ of alimentary canal is surrounded by peritoneum, so the outermost coat will be serosa. In that case, the connective tissue will be covered by simple squamous epithelium, that is mesothelium. But in esophagus, that is the connective tissue that is fused with the connective tissue of surrounding elements. If we are looking at the three-dimensional structure of various components of the alimentary canal, we can see the differences in their lumen, appearance of the lumen and the wall. First of all, here we are looking at the esophagus. In esophagus, we can see that lumen covered by stratified, squamous, non-keratinized. Here we are looking at the histological section of that part of the epithelium, that is stratified, squamous, non-keratinized epithelium. Next component here, that is the stomach, lining of the stomach. In stomach we will see pits on the surface, and it will be covered, so beginning from the stomach till the end of the anal canal. That region, all of this alimentary canal will be covered by simple columnar epithelium. Under the epithelium, we are looking at the lamina propria in here and here. That will be the small intestine, and it will have villi. Then large intestines, and then so that is stomach epithelium. Then that is component of the small intestine and the large intestine. And now we will focus on each of these components. First of all, that is the esophagus. Here we can see the cross section of esophagus. Esophagus, it is a tube with length of 25 centimeters, and it connects the pharynx with the stomach. In the lumen of the esophagus, at the rest state, we can see longitudinal folds which are formed by the lamina propria of mucosa and submucosa. These folds increase surface area, and during swallowing of the food, they disappear. These folds disappear, and the lumen becomes more. It will have more rounded appearance. Then, on the epithelium of the esophagus, as we're told, that is stratified squamous non-keratinized epithelium. Under the epithelium, we can see the lamina propria. The lamina propria is loose connective tissue, and in esophagus we will see here some glands that are cardial glands of esophagus, but they are present not throughout all of the length of the esophagus, but just in some regions. It is at the junction of the continuation of the esophagus, pharynx into the esophagus, and at the entrance of the esophagus into the stomach. When the esophagus where it continues into the stomach, they are the cardial glands of esophagus. They produce mucus. They are simple tubular glands. Over the lamina propria, we have muscularis mucosa, which composed of one layer of smooth muscle cells. Then submucosa. Submucosa, it is dense irregular connective tissue, and here we will see the submucosal esophageal glands. They present in submucosa. They are compound tubular alveolar glands, and here they include two kinds of cells. These are mucous cells, which produce mucus, and serous cells. Then we are coming to the muscularis externa of the esophagus. The muscularis externa of esophagus include inner circular and outer longitudinal layers of muscles. In upper portion of esophagus, they are present just skeletal muscles. In middle third of esophagus, we will see both kinds of muscles, smooth muscle and striated muscle. And in lowest part of esophagus, lowest third, we will see just smooth muscle tissue. Surrounding the muscular coat, we will see adventitia, that is connective tissue elements. Let's look at more high magnification of the esophagus. So here we are looking at the stratified squamous non-keratinized epithelium, lamina propria, then muscularis mucosa, and here number 5 indicates submucosa, which reach in esophageal glands. They present throughout all of the length of the esophagus. Then muscularis externa, that is inner circular and outer longitudinal layers. Then over the esophagus we will see the adventitia. Now we are coming to the stomach. Esophagus continues into the stomach. That is a junction between the esophagus and the stomach. Pay attention first of all to the epithelium of this region. So the stratified squamous non-keratinized epithelium continues into the simple columnar epithelium of the stomach. Under the epithelium, the lamina propria that contain cardial glands of esophagus. As we told with you that these glands, also cardiac glands of esophagus, they present just in two locations. That is where the pharynx continue into the esophagus and where the esophagus enters the stomach. In these two regions, these are narrowest portions of esophagus. Here present those glands, cardiac glands of esophagus, and we have to pay attention and remember that these are more vulnerable regions of esophagus which suffer from pathological conditions. They may be cysts, maybe hernia, maybe even cancer. They develop even mainly in these regions. We have to remember these regions. Also from esophagus, I want to know that esophagus has not its anatomical sphincters, but it has two functional physiological sphincters. First of all, upper esophageal sphincter, where the pharynx continue into the esophagus, and other one where the composed of the smooth muscle tissue that surround the orifice of esophagus, where it pierces the diaphragm and enters the esophagus. Also, it has external sphincter, that is the muscles of diaphragm, which surround the esophagus externally. And these sphincters are always on tonus, except at the time when the bolus passes into the stomach. Bolus, if you remember, that is partially chewed, masticated food that passes from the oral cavity and through the esophagus. This bolus will pass into the stomach. So these are the regions which we have to remember. Also, I want to know that in malfunctions or problems with the sphincters, this especially lower sphincter, esophageal sphincter, it may lead to returning to the entrance of the stomach, of the content of the stomach into the esophagus. And because of the acidic environment of the stomach which enters the esophagus, it will lead to burning feeling on that person, and it may be followed by regurgitation. This condition is called reflux esophagitis, and it may be possible in 15 to 20% of population. Now let's talk about the innervation of the alimentary canal. Before talking about the stomach, I want you to know about the innervation of alimentary canal. We know the layers of the alimentary canal, the tunica mucosa, submucosa, then the muscularis externa, and the outermost coat. You will have to know that alimentary canal, it has two components of innervation. First, it is intrinsic component, which is called enteric nervous system, and extrinsic component. Extrinsic component is composed of the sympathetic and parasympathetic innervation, and enteric nervous system, it is located in two regions in the wall of the alimentary canal. First component here, that is submucosal nerve plexus or Meissner's nerve plexus. Let's look here. These are nerve endings. These are nerves which are present inside of the submucosal nerve plexus or Meissner's nerve plexus. It controls the secretory activity of the glands in the wall of the alimentary canal and the motility of the mucosa. Then the next component, that is Auerbach's or myenteric nerve plexus or Auerbach's nerve plexus. It is present between the inner circular and outer longitudinal layers of the muscles. Here they are present. It's also called Auerbach's nerve plexus, and it is estimated that inside of the enteric nervous system there are as much neurons as in spinal cord. That's why some neurophysiologists think described that enteric nervous system as second brain even. So these are also, we have to know that they inside of the myenteric nerve plexus we have some cells. These are actually smooth muscle cells, but they are modified and they called causal cells, and it is thought to be they are pacemakers of the muscle contractility, muscle contractions of the wall of the alimentary canal. So as in pacemaker cells we can see in the heart wall, so the intestines also they have these pacemaker cells, and it was found that if we will cut the nerve fibers which come to this alimentary canal, it will continue its contractile ability, so peristalsis will be continue during some time without any interruptions. So our vagus nerve plexus will control peristaltic activity of the wall of the alimentary canal, mainly muscular contractions of the muscularis externa. So it will provide movement of the chyme in digested food along the alimentary canal. Enteric nervous system extends from the esophagus till the anus. So, and here we are looking that the extrinsic component of the innervation of the alimentary canal, that is formed by sympathetic fibers and parasympathetic. Parasympathetic innervation provided by vagal nervus vagus in most part of alimentary canal, except for the descending colon that is innervated by the sacral sacral region of the spinal cord. There arise sacral nerves. Parasympathetic innervation will control the motility, will control secretory activity of the alimentary canal components, but sympathetic innervation comes from the splanchnic nerves, and sympathetic innervation will control the lumen of the blood vessels, that is vasomotor. So activation of the parasympathetic nerves will increase peristaltic activity, will inhibit activity of the sphincters, and it will also activate functioning of glands. But sympathetic innervation will inhibit peristalsis and activate sphincter muscles. Now let's come to the stomach. Stomach, it is the dilated sac-like portion of the alimentary canal, and in its wall we can distinguish the same layers which is characteristic for all of the alimentary canal. So that is first one, tunica mucosa, submucosa, second one, third one indicate muscularis externa, and finally we will see serosa, that is covered by number 7, it is mesothelium. What we can see in the lumen of the stomach, these are, pay attention to this fold, they are rugae. If we will look at the internal surface of the stomach, we will see here the rugae. These are longitudinal folds that are formed by the lamina propria and the submucosa. They will be distended, they will be disappeared when the organ will be filled. Actually, at the rest state, the lumen of the stomach, it is approximately 50 milliliters content, but when we swallow and when during digestion, it may accommodate approximately 1,500 milliliters of chyme and the gastric juice. Then, in the lumen of the stomach, we are looking at the gastric pits. Gastric pits, these are invaginations of the epithelium that enter into the lamina propria. These are gastric pits. They increase surface area of the stomach. Here, look at gastric pits by more high magnification of the microscope, this light microscopy. And into the pits, into the gastric pits, open approximately 5 to 7 cardiac glands. The glands of the stomach, they located in the lamina propria. They are simple tubular glands. And Now we will look at these glands. Each of the gastric gland, all of the glands we can subdivide into three groups. These are fundal glands, cardiac glands, and pyloric glands according to their location. You know that stomach includes which parts? It has cardiac part, it has fundus, body, then pylorus. According to the location, so the glands we subdivided into these three groups. Here we can see that each gland, it has isthmus, neck region, and the base or the body. In the first of all, let's talk about the lining of the stomach, about the epithelium. The epithelium that covers stomach, that is simple columnar epithelium. But remember that each of these cells which covers the stomach, these are surface lining cells. Each of them, this is the secretor cell. It means that each of them produce secrete. The secrete is the mucus, but this mucus is called visible mucus. It will adhere to the surface of epithelium and it will protect it from the damaging effect of the gastric juice. Gastric juice, you know, it is acidic, pH there approximately 2. That's why mucus here, visible mucus, will protect the surface lining cells from the strong acid environment in the lumen of the stomach. Inside of this visible mucus, there is also also bicarbonate anions. These bicarbonate anions will buffer the acid. That's why, despite of the strong acid environment in the lumen of the stomach, on the surface of epithelial cells, we will have neutral environment. So now that the surface lining cells also continue into the glands, and in the glands we can see several types of cells. First of all, mucous neck cells. They are also mucus production cells, but they are different from the surface lining. They produce not visible but soluble mucus. Soluble mucus released onto the surface of the stomach, and it will cover the surface of the stomach, and it will be mixed with the chyme. This mucus will protect the wall of the stomach from the friction, abrasion. And then we are going to next type, the parietal cells. Parietal cells, these are largest pyramidal to ovoid shaped cells, which mainly present at the base and in the body of the gland. These are cells which produce gastric acid, hydrochloric acid, and intrinsic factor. Now we will look how this is produced, H-chloric acid. But before, let's look at other cells, heterochromaffin cells or DNES cells. These cells belong to diffuse neuroendocrine system. Argentophilic cells, also called, these are actually endocrine cells which scattered throughout the body, and we can find also in the wall of the alimentary canal. They produce various substances, histamine, gastrin, and now we will look at them also. Then chief cells. Chief cells produce pepsinogen. Pepsinogen will be converted into the pepsin in the acid environment of the lumen of the stomach. There are also gastric lipase produced and renin. So the three main types of enzymes are produced here. Then these cells, G cells, all of them belong to enterochromaffinic cells. Each of them have its own function, produce its own product. The lumen of the stomach, where we see the longitudinal folds here. Then the cells, mucous neck cell here. They have microvilli. Also, the microvilli present on the surface of surface absorptive cells, by the way. And the parietal cells, pyramidal shaped cells, chief cells, zymogenic cells which produce pepsinogen, we now know about it, and the endocrine cells. These endocrine cells, there are of two types of them. open type of cell which have connection with the lumen of the gland, and closed type. Both of them produce their hormones may produce into the blood circulation, into the blood capillaries here, or into the surrounding tissue. So they may have paracrine effect or endocrine effect, these hormones. Here we can see the parietal cell. We subdivided this cell into two parts. This is active part, what means active. The glands will be activated when during food ingestion, of course. So when we ingest food via nervous components, hormonal components, the glands of the stomach will be activated. And here we are looking at the active side of the cell. These parietal cells, they have differences from other cells that they have intracellular canaliculi. As you can see here, they have intracellular small canal. These are intracellular canaliculi. Into this canaliculi, we can see that open this microvilli. There are so much microvilli which increase surface area of the membrane, which is needed for location of several ion channels, pumps that are necessary for production of H-chloric acid. For the operation of those pumps, we have so much mitochondria here. They provide ATP for the functioning of these pumps. And in inactive site, we can see several tubular vesicular networks here. These tubules and vesicles, these are deposits of the membrane. When cell is activated, they form this microvilli, intracellular canaliculi are formed. Now let's look how the parietal cell produces H-chloric acid. First of all, the parietal cell, it has carbonic anhydrase enzyme. This enzyme will catalyze formation of, from the carbon dioxide, and from the water will be formed carbonic acid. Then the carbonic acid dissociates into hydrocarbonate anions and H protons. Then hydrocarbonate anion will be exchanged in basal cell membrane, that is basal part of the cell. In basal cell membrane, we have anion antiporter channel, that is anion antiport carrier protein actually. It will carry hydrocarbonate anions outside of the cell in exchange to chloride ions. So chloride ions will enter the cell through the basal cell membrane, and here you know that under the basal lamina there is connective tissue which reach in blood capillaries. So from the blood capillaries, the chloride will come and enter the cell through this via this anion antiporter channel. Chloride enters the cell. Then on the apical cell membrane, we have chloride uniporter channel, which will carry chloride into the lumen of intracellular canaliculi. Then it will enter the lumen of the gland. But here we know that it is dissociated into also protons, H protons, carbonic acid. So these protons will be carried via H potassium pump, that is ATPase. Remember that this ATPase, it is target for so many drugs, medicines, which nowadays we use to decrease acidity of the stomach. In treatment of gastritis, ulcers, that's widely used drugs. They are inhibitors of this pump. But what is doing this pump? This pump carries H protons outside of the cell in exchange to potassium ions which enter the cell. So the pump releases protons, and here we saw the chloride, and they then will join each other in the lumen of the organ. So H chloride is necessary for activation of the pepsinogen inside of the lumen of the stomach. The chloride, the potassium ions which carried inside of the cell, then they leave the cell through the potassium uniporter, which is present both in either in apical cell membrane or in basal cell membrane. Also potassium sodium-potassium pump operate here, so the potassium ions undergo recirculation. The enteroendocrine cells which present in the wall of the alimentary canal, including the wall of the stomach, they are of different types. For example, G cells which present in stomach, gastrin producing cells, then GR cells produce ghrelin. Ghrelin, it is the substance which will induce the sense of hunger, appetite, and perception of hunger, and also they will lead to receptional relaxation of the smooth muscles in the wall of the stomach. You know, the small stomach wall muscularis externa, it includes 3 layers of muscles. Will be inner circular, outer longitudinal, and additionally inner oblique layer of cells. It is additional smooth muscle layer. Then secretin producing cells, S cells. They mainly present in duodenum, but we can find them also in stomach. Gastric inhibitory peptide, motilin, they are all of these components produced by cells of enteroendocrine system. If we talk about the parietal cells, I want to mention that the activity of parietal cells will be stimulated by the action of gastrin, then histamine and acetylcholine. Acetylcholine released from parasympathetic nerve endings. Also, it is produced by special enteroendocrine cells in the wall of the stomach. And activity of parietal cells will be inhibited by this gastric inhibitory peptide, then somatostatin, it is also produced by enteroendocrine cells, and also inhibited by prostaglandins. Finally, the urogastron is produced by Brunner glands of the duodenum. They also inhibit activity of the parietal cells, so the production of the acid in the lumen of the stomach. That we have to know to be able to understand the regulation of the activity of the stomach. Here we are looking at the cardial part, and in next slide we will look at the pyloric part of the stomach. Here we have these glands, so the glands of the stomach, the cardial glands, and in pyloric part we will see pyloric glands of the stomach. They are similar to fundal glands, but what is the difference here? These glands include mainly mucus production cells. They also have surface absorptive cells, but the number of chief cells, parietal cells, if in cardial cells, cardial glands, they have in low in amount, but in pyloric glands, we even don't have them. And so here we have the submucosa, then the muscularis, and the serosal layers. Okay, small intestine. Let's now talk about the small intestine. It is the longest part of the alimentary canal. Its length may reach till 7 meters, and it has 3 portions: duodenum, jejunum, and ileum. In the lumen of the small intestine, we can see, first of all, these large folds. They called Kercrick. These are Kercrick valves. They composed of the mucosa and submucosa. These are folds. They form semicircular folds in the lumen, but they do not disappear during the passage of the chyme through the intestines, so they are permanent, and they decrease velocity of the chyme, and decrease the speed of movement of the chyme along the intestine. Is very important for proper digestion and absorption of substances. As you know, the small intestine is a region where all of the food products are completely digested and soluble and food substances are absorbed. If we will look at by more magnification at some region of the small intestine, we can see here villi. These are projections, leaf-like projections of the tunica mucosa, and here we are looking at the villi. Then epithelium forms invagination into the lamina propria. They called they will call crypts of Libercune or intestinal glands. All of these projections, invaginations, these folds are needed to increase the surface area of the intestine. That is very important for its proper functioning. Finally, let's look at one villus. If we will look at one of these villus here, we will see that the surface of the small intestine is covered by, first of all, it is simple columnar epithelium. Simple columnar epithelium includes cylindrical shaped cells. On the surface of these cells, we have microvilli. These are finger-like projections of each of these cells. So, these microvilli also will increase the surface area. Will increase surface area of the cell membrane. So all of these components will increase the surface area of the intestine by 300, 500 times. In the core of this villus, composed of the lamina propria, that is the lamina propria of mucosa. As we know, mucosa include epithelium, lamina propria, and lamina muscularis here, lamina muscularis. Inside of the lamina propria, we have blood vessels, blood capillaries, nerve endings, and the green-colored lymphatic capillary, that is the lacteal, that is very important component here, and it is needed for absorption of lipids. Here we are looking at the histological section of the duodenum, initial part of the small intestine, and that is jejunum. Now we will compare them. First of all, here, first component we know, tunica mucosa, submucosa, muscularis, and serosa. Here we are looking at the villi. Then, here also, in duodenum, in the submucosa, there is presence duodenal glands or Brunner glands. They are compound tubular alveolar glands which produce mucus. And also in submucosa, but submucosa of jejunum don't have those glands. Pay attention. And here present plexus of blood vessels. These are sections of blood vessels. Also, as we know, submucosa contain nerve plexus, Meissner's nerve plexus, and the muscularis externa. Here we have to know the cell. Pay attention to electron micrograph representation of the epithelium lining small intestine. The cells on the surface have this microvilli. They form striated border here. The cells have columnar shape, organelles, nucleus, basement membrane here, and lymphocytes may be present between these cells. And that is crypt of Libercune, that is bottom of the crypt or intestinal gland. Here we can distinguish cells which present here. And then the electron micrograph representation of the microvilli here. The cells joined to each other via specific intercellular junctional complexes. Now let's talk about the types of cells which cover the villi and crypts, that is lining of the small intestine. First of all, majority of cells are formed by surface absorptive cells. They are those cells which will perform final digestion of the food components and their absorption. Here we have also the goblet cells. Goblet cells are mucus production cells. Then we have regenerative cells in the wall of the small intestine on the surface of villi. Then we have also here the enteroendocrine cells, then microfold cells or M cells. They are cells which phagocytose the debris, the content of the intestine, and they phagocytose a particular matter, then convey them into the underlying connective tissue cells. Mainly, they will present them to the macrophages, and they will swallow the macrophages, which are antigen presenting cells. Then we on the bottom of the on the bottom of crypts we have Paneth cells. Here you can see which have acidophilic granules. They produce lysozyme antibacterial substances, and they are responsible for maintenance of microbiota, intestinal microbiota. They will kill pathogenic bacteria. Pay attention to these villi in the lumen. Inside of them we have blood vessels, then here lymphatic capillary and smooth muscle cells which located in the center. Because of the contraction, the intestinal villi change their position, and it's very necessary for contacting with the new portions of the chyme. It's necessary for digestion. About the regenerative cells, pay attention to the stomach, wall of the stomach, and wall of the intestine, small intestine. Here we can see the small stem cells, which located in the near to the isthmus neck of the glands, gastric glands. They undergo division and they move upward and downward. By that way, they replace the cells of epithelial lining. The rate of renewal here 4 to 7 days. But if we are talking about the intestine, these regenerative cells in the intestine, they are located near to the base of the glands, intestinal glands, and they will have unidirectional cell flow. They will move upward. The rate of renewal here approximately 3 to 6 days. That is faster than in the stomach. That is, now let's come to talk about the physiology. You know that small intestine is the region of final digestion. It is provided by enzymes because, first of all, enzymes which released by intestinal glands. Then you know that into the duodenum at the papilla of water open common bile duct, also pancreatic ducts. So both of them will carry necessary components for proper digestion. The bile and the pancreatic juice will be released into the lumen of the intestine by that way. And the activity of the pancreatic enzymes in the duodenum starts digestion, final digestion of the food substances. And then the formed disaccharides, dipeptides or dimers will come onto the surface of the intestine. Here we have these surface absorptive cells of the intestine. They have microvilli. As we know, the microvilli are covered by a thick layer of the glycocalyx that contain dipeptidases, disaccaridases. They will break these dimers till the monomers, and then they will be absorbed. Then they will enter the cell cytoplasm. Through them, they will carry to the basolateral cell membrane, and from here to intercellular space, and through the basal lamina, they pass into the lamina propria, and from there into the blood capillaries. But if we'll talk about the absorption, digestion of lipids, it has some specificities because the lipid digestion will need bile acids which will emulsify these lipids into small micelles. Then the micelles will be broken down by lipases into fatty acids and monoglycerides, also glycerol. The fatty acids with short chains, they will be absorbed into the cell, and and they will be absorbed into the blood circulation. But the fatty acids with long chains and as well as monoglycerides, they are absorbed into the cell. And here in the smooth endoplasmic reticulum, they undergo re-esterification, and there are reformed There are triglycerides. They will be triglycerides which are belong to the body, so these are components of the body triglycerides. Then they will be covered by the lipoprotein coat in the Golgi apparatus, and finally they will be formed kilomicrons. Kilomicrons through the basolateral membrane, cell membrane, will be released into the intercellular space, and from there to the basal lamina they pass into the lymphatic capillary. If you remember, on the center of the villi we have lymphatic lacteal. Through them the lipids will go to the lymphatic system, and finally they will be carried to the lacteal, through which they will return to the venous system. The endocrine cells which present in the wall of the intestines, they are also of different types. For example, PP cells, we have pancreatic polypeptide producing cells, L cells produce peptide YY, glycagon producing cells, L cells, D cells produce somatostatin, histamine produced by CL cells, then bombazine producing cells, vasoactive inhibitory peptide producing cells, enkephalin producing cells. Each of these components will have their own activity on the gastrointestinal tract. Large intestine, it has many similarities with the structure of the small intestine, all of the components of the layered wall. But what are the differences here? The large intestine, it is the main component, the main parts of it is cecum, colon, and rectum. It finishes at the anus. In the lamina propria, we are talking about the mucosa. The mucosa is on the surface of mucosa. We cannot see villi, but here we can see just intestinal glands, Libercune crypts. They are most numerous. And then the epithelial lining, it will contain mainly also surface absorptive cells, but in majority here we can see goblet cells. They are mostly more numerous than in small intestine. They will produce mucus that is necessary for not just lubrication of the surface, but also for the formation of the feces. So the chyme, from the remaining of the chyme, will be absorbed water, so the large intestine participate in absorption of the water and absorption of gases and formation of the feces for elimination from the body. Also enteroendocrine cells, they contain regenerative cells in the base and neck region of the intestinal glands. So they are the cells. Panet cells, they absent here. They cannot see them here in the large intestine. Then there's lamina propria of mucosa, and it is seen here. Submucosas, dense irregular connective tissue with vascular plexus, with nerve plexus. Then the muscularis externa, between them number 7 indicate myenteric Auerbach's nerve plexus that is present also here. Now let's look at the outer longitudinal layer of the muscularis externa. Here the cells do not form complete outer longitudinal layer, but they form 3 ribbons, the thickened region. They are smooth muscle cells accumulated in 3 regions, and they form tinea coli. And between the contraction of the muscles inside of the tinea coli will form the haustra coli, that is outposts on the surface of the colon. Outermost core, that is the serosa. The serosa will have fat projections, fat field projections that will called appendices epiploids. The appendix, it is the process, you know, that's extension of the cecum. The distinguishing feature of the appendix, it is the presence of large amount of lymphoid follicles in the lamina propria of mucosa. Finally, the rectum. Rectum, it is the final part of the large intestine, and it has the similar structure as in the rest of the colon. and the anal canal. Anal canal we can subdivide into three regions: colorectal zone, anal transitional zone, and squamous zone. Here we can see that in the wall of the anal canal there are present longitudinal folds, and they meet each other at the anal valves. Between anal valves we can see anal sinuses and also anal columns. The region of the anal canal that is above this region, this region is correspond to pectinate line that is covered by simple cuboidal epithelium. The region between these anal sinuses and external anal orifice is covered by stratified squamous non-keratinized epithelium, and finally in anus it will be stratified squamous keratinized epithelium, that is the skin actually. And also in lamina propria of mucosa, here we will have anal glands and circumanal glands. And in submucosa, above this pectinate line, we will have extensions of capillaries. They will form superior hemorrhoidal plexus, and at the region of anal canal, we will have inferior hemorrhoidal plexus. So that's all for today. That's end of our lecture. Thank you for your attention.